Kemango is a 3-year training project implemented by IRD, collaboration with 3 Vietnamese universities: Hanoi National University of Education, University of Science and Technology of Hanoi, and Ho Chi Minh University of Science. The second edition of this training was located in Ho Chi Minh University of Science and Can Gio Biosphere Reserve. Theoretical courses were held in class at the beginning of the course at University of Science. They aim to introduce the metabolic diversity of plants, the way in which the metabolites produced by plants are then released into the environment, and the effects. After 2 days in class, we went to the Can Gio Biosphere Reserve in order to work directly in the mangroves. Students are put in conditions to take forestry measurements. They can also experience working with their feet in the mud, accompanied by the inquisitive local fauna. At the reserve's botanical garden, students are comparing the growth of 3 different mangrove species. To do this, they measured the height and diameter of all the trees in 3 plots of 10 meters on each side. They also recorded the species richness, meaning the number of species identified in each plot. Back in the laboratory, they recorded the results of this experiment. To approach techniques used in chemical ecology, they learn how to sample volatile organic compounds emitted by plants. These compounds are the smell of the plants produced by leaves or flowers. They are particularly important in communication between plants and their environment, such as insects for pollination or other plants. A branch is enclosed in a dynamic chamber where airflow is constant. Compounds are trapped in tubes, which will be analyzed in the laboratory using gas chromatography system. Volatile organic compounds sampled in the field and analyzed in the laboratory are identified in order to determine the smell-producing compounds in plants. Back to the laboratory, they recorded the results of the experimentation they set up before going to Can Gio. They measured lettuce seedlings used as a laboratory model species that had been watered with different concentration of aqueous leaf of 2 mangrove species. But this experimentation can answer to the research question: can mangrove aqueous extract affect the growth of a target plant species? They carried out statistical treatments of all the results acquired during those days of work.
This useful and solid knowledge that I gained from the Kemango training program that make me confident to develop my scientific and academic career, especially plan to apply the PhD in the future.
So in the past, I used to know that the tree can communication with other tree, but I don't know how they can. But now I understand the mechanism of them, and they can produce compound to talk, to defend with other environment factor.
The students here are really enthusiastic. They ask a lot of questions. They make a good job on the field, but also here in the lab. They are always with us, so we have a good relationship.
So great to work with students who are so motivated, and you can really see them, work hard, and they ask a lot of questions. So that's good, it's rewarding for us, but it's also challenging for us.
I want to say thank you to all of teacher and staff who hold the score. They try to prepare a good schedule and also lesson for all of students in the course.